Hello, can you identify this molecule? This is DNA or deoxyribonucleic acid. Inside cell, you will find most commonly double-stranded DNA in which two strands are intertwined to form a double helix. This is called B form of DNA. Here is the timeline of history of genetics. Starting from the great work of Mendel, we have reached to this point the experiment which proved that DNA is the genetic material. Although discovery of DNA predates to the 18th century, but DNA was ignored for a long time and it was a protein that got a lot of attention. F. Griffith, British bacteriologist, in 1928 performed transformation experiment with bacterium Streptococcus and he discovered transforming principle and he assumed that this is genetic material. However, Hershey and Chase finally established that DNA is genetic material. The next step is to understand the structure of DNA biopolymer and to know that what are different constituents of DNA. The DNA molecule was discovered in early years of 1950s, but remember it was not a molecule, it was a structure. During the early years of 1950s, an English chemist named R. Franklin started to work on DNA molecules. She isolated DNA crystals and exposed to X-ray. She obtained X-ray crystallograph pattern of DNA molecule that triggered the idea that DNA was a double helix. Around the same time, Watson and Crick worked on the structure of DNA molecule and reached a similar conclusion like R. Franklin did. Watson and Crick shared Nobel Prize with Wilkins for their discovery of double helical structure of DNA molecule. According to Watson and Crick, DNA molecule is made up of three components. One is pentose sugar molecule called deoxyribose. How it is obtained? It is obtained by removing one oxygen atom from the ribose molecule. Thus, the name of DNA is deoxyribose nucleic acid. The second component is a phosphate group. It binds two adjacent sugar molecules together. The third and fifth carbons of adjacent deoxyribose molecules bound to a phosphate group. Lastly, the nitrogenous base forms the core of helix. These are four different nitrogenous bases that bind in pairs. These two are called purine and these two are called pyrimidine. A purine binds with a pyrimidine. Is any purine can bind with any pyrimidine? No, it is thumb rule that always adenine binds with thymine and guanine always binds with cytosine. These molecules are held together with the help of hydrogen bonds. Adenine binds with thymine with the help of two hydrogen bonds, whereas guanine binds with cytosine with the help of three hydrogen bonds. Did you know one unit consisting of a molecule of deoxyribose sugar A molecule of base and a molecule of phosphate is called nucleotide. What if a phosphate group is removed from a nucleotide? It is nucleoside. In a nucleotide, there is phosphodiester bond between a phosphate group and deoxyribose sugar, whereas glycosidic bond links a nitrogenous base to the sugar molecule. DNA molecule has four different types of nucleotides. These nucleotides are distinguished by their nitrogenous bases. DNA molecule is a biopolymer. Polymers are larger molecules built up by linking smaller molecules called monomers. For example, carbohydrate is polymer having glucose monomers. Similarly, protein is polymer of amino acid. DNA is made up by linking nucleotide monomers in a non-random sequence. Several nucleotides linked in a predefined sequence with the help of phosphodiester bonds form one strand of DNA double helix. Each chromosome has two DNA strands. These two strands run anti-parallel, means 5 end of one strand is parallel to the 3 end of other strand. The nucleotides forming each strand of DNA are connected by hydrogen bonds. These hydrogen bonds happen between two bases. Along the length of DNA molecule, there are alternating major and minor groups. Asymmetrical spacing of sugar phosphate backbone generates these groups. These groups are used for binding of proteins to DNA molecule. The diameter of helix is 2 nanometer. One major group and one minor group form one helix. The length of one helix is 3.4 nm. There are 10 base pairs in one turn of DNA helix. The two base pairs are placed at 0.34 nm distance in one strand. In next lecture, we will discuss about the DNA packaging in eukaryotic chromosome. Thank you.